Before we begin to analyze new enzymes, let's first recap what we know. First, we know that in Alzheimer's disease, there is a decrease in glucose metabolism in the brain. Second, this is characterized by an increase in phosphofructokinase and a decrease in both pyruvate dehydrogenase and cytochrome C oxidase. Together, these results contribute to the increase of amyloid beta plaques characteristic of Alzheimer's disease. Moving on to examine the other enzymes, we'll look at the results of Bigl et al. In the study entitled Activities of Key Glycolytic Enzymes in the Brains of Patients with Alzheimer's Disease. This research was focused upon measuring the activities of specific glycolytic enzymes, then furthering that research in determining if there was a correlation between this activity and the activation of astrocytes in Alzheimer's disease. Before we can explore these results, we need to take a step back and examine astrocytes. Astrocytes are star-shaped glial cells with a supportive and regulatory role with neurons. These astrocytes express glial fibrillary acidic protein used for the detection and quantification of astrocytes. Specifically in Alzheimer's disease, an increased number of reactive astrocytes is an invariant feature of the pathology of the disease. So let's take a look at what we already know. It's already well established that there is a decrease in neuronal glucose consumption in Alzheimer's disease. However, the reason behind this decreased glucose consumption is unclear. Is this a limitation of metabolic capacity, unable to metabolize as much glucose, or does the diseased organ require less glucose? Mechanistically, is this a functional impairment, unable to metabolize glucose, or is it due to the loss of neurons? This now brings us to the theme and overarching purpose of this study, to determine the cause of the reduced glucose metabolism in the Alzheimer's disease brain. To study this, researchers used post-mortem human brain samples. Samples were dissected from specific locations within the brain and either homogenized for quantification or fixed with paraformaldehyde for immunohistochemistry. To move into the enzyme results, we can first look at hexokinase. Hexokinase catalyzes the first step in glycolysis, the phosphorylation of glucose, as seen in this diagram. There were no significant increases in hexokinase levels, but the general trend appears to be a slight increase in activity within the cortex areas of the brain. This is consistent with the increase in PFK, since if PFK is increased, there should be somewhat of an increase with its precursors. Thus, if more samples were taken, this may have reached significance. The next enzyme we're going to look at is pyruvate kinase. Pyruvate kinase catalyzes the final step in glycolysis, converting PEP to pyruvate. There was a significant increase in pyruvate kinase activity in the frontal cortex, shown by the bars outlined in red. The increase in pyruvate kinase may serve to counteract the decreased glucose, thus allowing more of the limited glucose to be metabolized at one time. The final enzyme we're going to look at is lactate dehydrogenase. Lactate dehydrogenase catalyzes the conversion of lactate into pyruvate and vice versa. In doing so, the pyruvate can then be used in the metabolic cycle within the brain. It was found that the enzyme activity and the protein levels of lactate dehydrogenase were increased in the frontal cortex shown in red, while in the basal forebrain, only enzyme activity was significantly increased. The frontal cortex had an increased level of GFAP, indicating an increased number of astrocytes. Furthermore, the increasing GFAP correlated to the increasing severity of Alzheimer's disease and the increasing PFK, suggesting that astrocytes may contribute directly to Alzheimer's disease. Combining the increased lactate dehydrogenase and the increase in reactive astrocytes, researchers developed the theory outlined here, that it is the reactive astrocytes in the brain that are consuming the glucose rather than neurons. These astrocytes are then producing lactate, which is being consumed by neurons. Thus, the neurons must increase lactate dehydrogenase in order to convert the lactate into pyruvate for metabolic processes. From these findings, researchers suggested that the elevated enzymatic activity suggests a metabolic alteration hindering glucose consumption, not a reduced need. Mechanistically, this suggested the lack of neurons doesn't cause the reduction, but rather the impairment of neuronal glucose consumption by astrocytes. In summary, researchers found that two significant yet opposing processes occur in these brains, one being a reduction in neuronal glucose metabolism, the other an enhanced glial activity, which may be compensation in providing alternative fuels to neurons.